Hello students, welcome to George Biology. We will start with semi-autonomous cell organelles. Now if you talk about the semi-autonomous cell organelles, in the name itself you can find the definition. Semi, that means half or partial. They are half or partial autonomous with respect to their functions. Due to presence of their double-stranded circular DNA, they can perform their activities. Right? It means they are not completely dependent on the cell. They can do their own functions due to presence of their own DNA in them. So called as semi-autonomous cell organelles. So why they got that property? Due to presence of double-stranded circular DNA. Circular DNA. Remember this point? Double-stranded circular DNA. Based on this one, we'll try to connect the origin of the semi-autonomous cell organelles in the next part. Now, under semi-autonomous cell organelles, there are two aspects which we have to study. Number one, mitochondria, and number two, plastids. We will start with number one, mitochondria. Now, if you look at mitochondria, they are variable in their shape, size, and number. They can be sausage shaped, they can be cylindrical in shape, and the diameter that can range between 0.2 to 1 micrometer in diameter, and they can range in a length of 1 to 4.1 micrometer in length. If you talk about mitochondria, until and unless if they are not specifically stained, they are not visible under the microscope. It means what is a stain that's specifically required to study mitochondria? A stain which is required, that is Janus green. Janus green. And what kind of stain it is? It's a supra-vital stain. That means supra, that means specific. Vital, that means in living condition, right? By using this stain, we can study the mitochondria when it is in living state itself. So we call it as a supra-vital stain. So if a question is asked, the supra-vital stain that is used in the study of mitochondria, that is Janus green. Okay. Now this mitochondria that was first reported by, it was first reported by Kolliker. But the term that was given by Benda. But before taking the term of mitochondria, this was earlier called as bioplast. So mitochondria, they are called as bioplast by Altman. And the term mitochondria that was given by Benda. Now, if you observe the structure of mitochondria, that consists of 4 regions, and these 4 regions that includes 2 compartments and 2 membrane systems. On the left side, we'll take the compartments. On the right side, we'll take the membrane systems. So it consists of 4 regions. So this is the outer membrane, number 1, outer mitochondrial membrane. And this outer mitochondrial membrane, it is rich in porins. As it is rich in porins, it is permeable in nature. It is permeable. And it is smooth in texture. It does not consist any kind of infoldings, due to which it is smooth in texture. And it consists of 60% proteins. And along with that, it consists of 40% of lipids. Number 2. Apart from this one, it also consists of certain membrane at the inside, and this membrane, it consists of infoldings, and these infoldings that helps in increasing the surface area of the inner membrane, and that inner membrane, it is called as inner mitochondrial membrane, IMM. It is rough in texture, as it is consisting of many knob-like structures called as F0, F1 particles. It consists of 80% of proteins and 20% of lipids. But it consists of a special kind of lipid, and this special kind of lipid, that is called as cardiolipin. Cardiolipin. And this cardiolipin that makes this membrane almost impermeable. It means it makes the membrane selectively permeable in nature. selectively permeable nature. So if a question is asked on comparison between outer mitochondrial membrane and inner mitochondrial membrane, which is more permeable, that is outer mitochondrial membrane. Now this in the mitochondrial membrane, we said that it consists of infoldings, and this infoldings, they are called as cristae. Cristae. So these are the two membrane systems. We'll go with the compartments. So here if you're observing the two membrane systems, it has divided the inner region into two pieces. The region between outer membrane and inner membrane is called as intermitochondrial space, and the entire inside, that is called as matrix region. So it consists of two regions. I'm taking this as a continuation, number 3. So the region which is present over here between outer mitochondrial membrane and inner mitochondrial membrane, that is perimitochondrial space. Perimitochondrial space, or else it can also be called as intermitochondrial space also. And the last one, the inside region, the most important part, that is matrix region. Now this matrix region, it consists of dense homogeneous semi-fluid material or substance that is called as matrix. Number 4 is a matrix region. And what is this matrix region? It's a homogeneous semi-fluid substance. It's a homogeneous semi-fluid substance. Now what does this consist? It consists of a double-stranded circular DNA. So it consists of double-stranded circular DNA. So here I am taking that. Consists of double-stranded circular DNA. Along with that one, it consists of some amount of RNA also, and it consists of 70S ribosomes. Now, as they consist of 70S ribosome, it acts like a site for protein synthesis. It helps in protein synthesis. So what are the things which we have studied? It consists of 4 regions, 2 are membrane systems and 2 are compartments. In that compartments, which is a major ones, that is matrix region. Now we said that it consists of infoldings, and these infoldings, they help in formation of cristae, and this cristae is rough in texture, we said, and this rough texture is due to presence of knob-like structures. They are called as oxysomes or else F0 F1 particles. So here in the diagram you can see. So what does this consist? It consists of cristae. Over the cristae, it consists of knob-like structures, and these knob-like structures, they are called as oxysomes, or else it is also called as F0 F1 particle, or else it is also called as Fernandez and Moran particles. As it was first observed by Fernandez and Moran, we have given the name as Fernandez-Moran particles. Now if you look at this oxysomes, they are knob-like structures which consist of head and tail. So here a simple structure will take. So here is a F0 F1 particle. The lower region, it is called as a base, and the middle region is a stalk-like structure, it's a slender stalk-like structure, and over the top it is called as a head. Or else it can also be written as the lower region as proton tunnel F0 particle. Now this one acts like a proton tunnel where H plus ions will be traveling through this and hits the head part, and that starts rotating and helps in combining ADP with inorganic phosphate and helps in formation of ATP. It means if a question is asked, what is the site of production of ATP? That is F0 F1 particle. Exactly which part? That is F1 particle. And to which region this ATP is getting released? That is matrix region. Now this F0 particle through which H plus ions are traveling, that is called as H plus tunnel. And this head region, that is called F1 particle, and this F1 particle helps in ATP synthesis, so it acts as a ATP synthase particle. Let's go with the functions of mitochondria. Functions. It helps in aerobic respiration, where in the aerobic respiration, there are 4 steps that are present. That is glycolysis, link reaction, Krebs cycle, and ETS. Out of all, 3 steps that takes place in the mitochondria itself, except glycolysis. It means it acts like a site for Krebs cycle. And link reaction and ETS also. Why? Why? Because the enzymes which are required for the Krebs cycle, they are present inside the mitochondria. Now, if we talk about inner mitochondrial membrane, it acts like a site for ETS and oxidative phosphorylation. Now, by performing this ETS, there will be production of cellular energy that takes place. It helps in cellular energy production. And this cellular energy, it is in the form of ATP. As it helps in ATP production or else cellular energy production, that is why they are titled as powerhouse of cell. Powerhouse of cell. So what is a cell organelle that is called as powerhouse of cell? That is mitochondria. Let's go with the next one. It consists of enzymes for performing the Krebs cycle, and it contributes 80% of total cell enzymes. It means major production of enzymes that are produced where? In the mitochondria. Let's go with some special properties. Number 1. If you look at the maize plant, in maize plant, it consists of certain gene in the mitochondria that is called as CMS gene, and this CMS is abbreviated as cytoplasmic male sterility gene. So what is this one? It's a cytoplasmic male sterility gene. Cytoplasmic male sterility gene. It means what happens with this one? If this gene is present in the male, now that male becomes sterile. If this gene is absent in the male, then that is fertile. If present, sterile. If absent, fertile. Now this is the property of CMS gene with respect to the maize plant. Let's talk about one more special property. Now this special property, it represents how this mitochondria gets inherited. It gets inherited from the cytoplasm of the egg cell. Egg that contributes high amount of cytoplasm to the zygote formation. Through that, the mitochondria will be transferred. It means what kind of inheritance does this mitochondria shows? It shows maternal inheritance. It shows maternal inheritance. In genetics, we will get a connective questions from this topic, right? What are those questions? Give an example of maternal inheritance, that is mitochondria, or else mitochondrial inheritance that come under maternal inheritance. So this is all about the properties of mitochondria. Here you have to remember one thing. Twice we have got a question in mitochondria, in 2019 as well as in 2016, where the question is based on the function. That is, what is the site of ETS? The site of ETS is inner mitochondrial membrane. In the next session, we'll go with a small subtopic, that is endosymbiotic origin of mitochondria.